who they are.
Another area where science is making major advances, human genetics. Crispr, a kind of genetic scissors, has been around for a long time, but researchers are using it in a new way to change the genetic code in cells to fight cancer and other diseases. Here's Donna Hanover.
CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. It's a bit of a mouthful, but the easiest way to think about CRISPR is it's a technique that we can use to change DNA, to edit DNA. That's what it is at its very, very heart. And the reason that we might be interested in editing DNA is when we find, through, say, sequencing studies, we find a mutation that we think has something to do with the disease, like autism, cancer, diabetes. What we might want to do is really test to see, does this mutation cause this disease? And so the CRISPR tool is a wonderful way for us to very efficiently inactivate genes and test. If I inactivate gene A, does it cause cancer? If I inactivate gene B, does it cause MS or Parkinson's?
Lead investigator Dr. Neville Sanjana at the New York Genome Center says it's like a scissors to cut out and change small sections of genetic code in cells in the lab. How does it work? The CRISPR system commonly used is known as Cas9. It has an enzyme that's a short piece of DNA protein and an RNA molecule.
The RNA component of the CRISPR system guides the protein component, the scissors, to a specific place in that 3 billion base pair genome and tells the protein where to make the cut.
How do researchers physically accomplish this?
It's a little bit of liquid that we deliver into the cell, and either using an electroporator like that, we can shock the cells and make little holes in them to deliver it in, or we can bind the DNA together with lipids, fats, because the cell membranes are made of fats, and so if we bind it together with some fats, that tends to be drawn into the cell. We create the CRISPR constructs here in the molecular biology wet lab, and then we go into the cell culture room and deliver these into cells. And then after a few days, to validate that the CRISPR actually worked, we take the DNA out of the cells and we visualize that DNA using something like the gel electrophoresis you're going to see. It uses charge, DNA is a charged molecule, to separate DNA by size. So what happens is that bigger molecules of DNA, longer molecules of DNA, don't run as fast through the agarose gel. But smaller pieces of DNA run much faster. And that's why you see these bands in one lane running down the gel. And the two bands reflect that the editing process has happened.
And how do researchers know what the change in a cell means?
We look at things like, how does the cell grow? Does it proliferate out of control? This is the kind of very clear phenotype that we associate with cancer, a tumor that can't stop growing.
Dr. Sanjana says a great aspect of CRISPR is its programmability, meaning researchers can change thousands of cells in different ways at the same time. Dr. Sanjana has used CRISPR to study genes that cause cancers to metastasize to other parts of the body. He also studies melanoma to find out why drugs used to treat it may work initially, and then genes cause some patients to develop resistance.
And so CRISPR has really been a breakthrough for this.
In his work on autism, Dr. Sanjana uses CRISPR on healthy cells to cut out small sections of DNA and replace them with mutations suspected of causing the disease.
Once you've made the cut, how do you put in something new?
After CRISPR makes its cut, there's a bunch of machinery in the cell that wants to repair any chromosomal break. And so if we deliver a small repair template, the cell's natural repair machinery will actually use it to insert that piece of DNA, changing the DNA from its original form to the edited version that we want to put into the cells. A lot of what we've done in the pre-CRISPR era has been just observation. We look to see when we see an organism or a human being that has a particular disease, we maybe look at their genome, we read the DNA. But what's been lacking really has been the ability to write the DNA.
What does the future hold?
Gene editing is really just 50% of the task. The other 50% is finding ways to analyze the cells we create. We need to be able to analyze these cells and say, after we create the mutation, do the cells look like the kind of cells that are involved in diabetes or cardiovascular disease or in autism or in cancer? And really the quantum leap forward with CRISPR is that it's now quite easy to make the mutations. But still that second step, seeing whether the cells actually have something that looks like the disease, that's still really quite a challenge.
Meanwhile, while genetic research has largely focused on the 2% of the human genome that codes for proteins and which is familiar to most of us, Dr. Sanjana says the part that doesn't make proteins comprises 98% of the genome, and these regions are also involved in disease. His lab is developing new CRISPR tools to explore how this part of the genome works in pursuit of more answers for medical research.
I think I'm just incredibly excited by the confluence of biology and technology in this field, which I think is a fantastic thing to have the opportunity to work on things that can affect people's lives in such a positive way.
So as genetic scientists improve and expand their use of CRISPR, hope is rising. In the fight against some of the most intractable diseases in human history, they have a tool that may finally help us win. I'm Donna Hanover for Science and You.
When we talk about the environment,